then be more easy. No, I'm just joking, but I hope that someone do some questions about the presentation, about what you can explore, the intersection with everything that you saw yesterday and today. I will continue the discussion, but today using another spectroscopy. And I will show at least 3 examples that you can use this. And I hope that this brings some ideas to your experiments. Okay, then the topic today is fluorescence spectroscopy. The fluorescence is the emission of light by one substance that has absorbed the light or another electromagnetic radiation or different waves then affect this material, how I discussed with you yesterday. Then, who started these studies was George Stokes with the phenomenon in 1852. The name was derived from the mineral fluorite, the calcium dichloride. Then for this, that is fluorescence, come from this, the origin of the name. Then I just do one, like a frame about one of stages of the material. I won't discuss the levels of energy, but I would like just to look for a specific step of the shelves when you have the electronic distribution. Then you can, if you look for the last level that you that electrons are organized in the different levels of energy. In this level, you can name it like ground state, that is like that is the ground state. And another state is the excited states. Then, if you look this specific state that you have the 2 spins, like we wish that be organized in the parallel, but in different ways. Then this is the ground, the fundamental in your last distribution level. Then but you can excite this, and the electrons can be in 2 different states. If the electrons organize you excite, then go to the up level, but you maintain the anti-parallel directions, and you can organize this in 2 different ways, or you can excite the first spin, or you can excite the second spin. Then for this, that is singlet, is the double state that you have here, that the spins can organize in anti-parallel direction. But if the electrons go to the another way, but no in the parallel, anti-parallel level, but you can be in the same directions. You have one organization, and you can organize these electrons in another way, that you have the buff in up to the down, or you can organize where you have you have 3 different ways to organize this, with 2 in the same direction, or in the down, the up to the down, or one with the up to the down, up to the down. Then it's different from here. Then you can obtain 3 different ways of organization of your electrons. For this, the name is triplet. Then from the ground to the excited levels, you can organize the electrons in 2 different ways. One, where you maintain the parallel and anti-parallel, or in this case that you have, for example, here, one is up to the down, and another is up to the down. Then it's completely different from what you obtain to the fundamental level. Then you can organize the electrons in 3 different ways. Then for this, that is triplet. Then is this, that is I write here. Then let's go to distinguish these levels of energy. Then the ground state, the excited state is like I said to you. Then you have the fundamental level where the spins are in the same level, but you can excite this to the up. Then you can have one in each level, in the singlet or triplet organization. Then the singlet, all spins are paired, and in the triplet state, you have the parallel spin in each one in a different direction. And why this is important for us? Because how your electron be excited and emission, you have different phenomena. Then, if different paths and which excite these electrons. One path will result in the fluorescence. Then, just in one specific case, when you start from the ground level, you go to the excite level, you have the phenomenon of fluorescence, because you have another phenomenon like phosphorescence or just absorption. Then I would like that you have in mind that we don't have just that first singlet level that I showed you. You have another levels. You have the first, the second singlet, depend of the level of energy that you inject in your system. But to the fluorescence phenomenon happen, you need to be involved between the first singlet excitation. For this that I just look for this frame, just for this moment. But when you excite your electrons, you have another states of singlet. But it's important for fluorescence, it's just the first, like here, the first singlet level, no, the second. Then the phenomenon be different between the phosphorescence and the fluorescence is just the time that you return to the ground level. Then if this time is very quickly compared to the this another phenomenon, then you have the directly emission of the light. What this means? That between the singlet, to the conversion to the plate level, you have like the electron change the emission the light go to the another state of the organization. Then to have the phosphorescence, you need to be involved the triplet organization. But when you have the fluorescence, you don't have any triplet organization of the electrons, then they go directly from the singlet to the ground state again. Then, is this. The molecules in the fluorescence emission process has the lifetime that is shorter than compared with the triplet. Then to happen the triplet, you need the conversion, for example, the singlet to the triplet emission the light. Then the fluorescence go directly from the singlet step. Here, it's just the same picture, but now just to evidence what I am talking. Then here, when you excitation from the ground, you go to the singlet. If you emission a little bit of energy, you go to another state, you go to the triplet, and then triplet emission, the phosphorescence process. Then if you have the singlet, you go direct for the ground effect, you have the ground state, then you have the fluorescence. Then now, just to be clear about the terminology, I will discuss the specific names. If you look for all the phenomena, you have the luminescence. The luminescence is the process that involve any absorption of the light and in the emission. Then luminescence involved, if you look in this figure, here. Involve not just the fluorescence and the phosphorescence, but the absorption that go to the up levels, like the second singlet level. Then the luminescence involve all energy transitions from your electron. Then if you go in then this is the mean of luminescence. But the photoluminescence is the generation of luminescence through the excitation of the molecule, but involve the just the singlet level in case of the fluorescence, and the triplet in the case of the phosphorescence. Yeah. No. Here. Then, just to fix a little bit more, to start to discuss the technique, this is the is come to the last slides about the theory that I would like to discuss. Then, just to weight the same process in a different figure. Then, the absorption process start here from the ground level. It go to the up where you have the excitation. But another point that is important to be in mind about the fluorescence, that the fluorescence just happens when the electrons are in the lower level from the singlet step. What this means? That you have here the singlet step, but you have different levels of energy. But to happen the fluorescence, the electrons need to be in the last level from the excited step in the singlet. It goes direct from the ground level. Just remember the energy and the Planck equation that you have to the correlation, the frequency, the wavelength speed. Yeah. Then I'm very emphatic to show the same thing many times, just to be in mind. Then the excitation and the transition between the levels in the emission of the light. Then the base of the fluorescence involve this, but during the process of excitation, how I already said, you come from the ground effect, it do excited by electrons, but you have to the vibration, then you have a different phenomena that be involved, and the relation, the relaxation that when you emit the light is the process that you electron go from a level to the other. But this is very important because if you have a transition step between the singlet to the triplet, this vibration, then you emit some energy, but not enough to go to the ground level, but you have a transition step. In the emission is just the return to the ground level. Then, about the fluorescence, which kind of probes or molecules you can observe this effect? You can observe this fluorescence effect like intrinsic fluorescence that is evident in some amino acids like tryptophan that naturally can suffer this process of excitation, but you have a lot of different probes, molecules that you can use for analyze some materials that can interaction, and you can observe this effect. Here, I would just like to expose some applications to the fluorescence, like use this to quantify some compounds that can bind to some fluorophores. Then you can use this to quantify or to qualify some molecule. You can use this for assay experiments like the vitamin B, hormones, carcinogenic compounds. Then you can bind this fluorophore with some molecule of interest to evaluate that the presence of that cells. You can use these probes for microscope studies. Then you can bind these some probes with a specific organelle or some protein that you would like to study, like in the FACS experiment that I showed some example for you about the use of the fluorescence to protein-protein interaction studies. Or you can look for the protein structure. If you look for some intrinsic fluorescence, then you can look how the your protein suffer with the increase of the temperature, then you can just look for some amino acids in the time and see how the fluorescence happen when you increase, and or to the kinetic experiments. These that I highlight in red, I show some examples for you. Then the fluorescence decay is just the one way to estimation the lifetime of the fluorescence. When you are work with some probes, like in the FACS that you use one specific fluorescence molecule to attach to your target, this lifetime is so short. The lifetime is the time of the decrease the fluorescence effect. But what I would like to say that about the shelf or the experiment time that you can use that probe. That probe means that it can excite emission, but it can do this a lot of times. Then you can look one experiment in the time. No, it's just you excite emission, you finish the process. You can excite again the emission, but this process has some time. But this isn't the lifetime. The lifetime is the time that you observe the maximum emission, it decrease. Another is like the life, is the term is, I'm talking about two different lifetimes. One is the emission, another how many times you can use that probes. There's different times. But here is about the time of emission. You can come. Okay. Then you can correlation this absorption, the number of molecules we can excite. Then the fluorescence intensity is proportional to the number of excited molecules. Then is like the obvious, but it is important to see that you can correlate this number of molecules in the time, and that this number of molecules is directly correlated to the intensity. Then you can rewrite this equation like d intensity in the time, like minus k to the intensity to the time. Then you can directly correlate the number of molecules with your intensity. Then this means the decay of excitation molecules in your experiment from the first order the process. Another term that is used in fluorescence is about the quantum iode. That is the correlation. What is the quantum iode? Is when you absorb, the light is absorbed, just one fraction of this emitted by fluorescence is absorbed, another fraction no. Then is the correlation between the quantity emitted of fluorescence by the quantity of absorbed of the light. This correlation time is the quantum iode, and it can correlate to like the tau, is the lifetime of all molecules excited by the tau fundamental that is correlated with the intrinsic lifetime of the fluorescence. Then the fluorescence intensity is proportional to the product of absorbed. Then you have more fluorescence depending on your probe. If it absorbs a lot, you can emit a lot. This is directly correlated. Another term that I won't explore a lot, but I just show is about the fluorescence resonance transfer, that the name is FRET. That is when you have the superposition of the different probes has the absorption and emission light, because depend of the characteristic of one probe, we can excite it with a wavelength, but it emits a different wavelength. Then if you use different probes, but for example, one absorb is started to the emission exactly or around the wavelength that another probe started to absorb. Then you have a superposition between the excitation of one probe with the absorbance of another probe. Then when you have this kind of intersection, you have the like a resonance process that the name is FRET. Then the transition the overlap of the emission of a spectrum of one donor is correspond to the is this, that we have the overlap of the emission of one aspect donor, then you have one is like one donor of the energy, and another absorb that. It's like that you have it's like that you have two sources of excitation, because naturally you need to excite your probe, then you do this excitation. But in the same time, the emission from the another probe works like another source of excitation. Then you have two excitation in the process. You can correlate these two with another efficiency process that you have one specific equation. Then the FRET is a great method to determine the distance between two fluorophores. Then this is important if in some experiment you would like to see the distances, then you can use the FRET to obtain some information about your molecules that are in interaction. Or if you use some probes with DNA, then you can obtain some information about the distance to using the FRET. But now I go direct for the fluorescence, exactly, and they use with that three examples that I said. Anyone would like to do some question? No? Okay. Sorry.
Since the transitions always happen for the lowest cyclic state, is it right to assume that the fluorescence will be mostly located in the IR part of the spectrum?
Just repeat, because I didn't listen very well.
You mentioned that fluorescence is a phenomenon that occurs between the lowest singlet towards the ground state.
Yeah.
This is low energy. Is it correct to assume that most fluorescence happens around the IR, the infrared region? Like
yeah, exactly. Yeah. Okay. Sorry. Then what's the instrumentation that you need for the fluorescence? You need the one source of energy, then this excited, you have it, in this you have two different monochromators that monochromates to the light. Then you have one involved with the excitation, then you need the direction, the light that you need to the excitation your sample. What I'm talking about excitation in monochromator. How I said to you, each probe has one specific wavelength to excitation. Then you need one monochromator that is just select one specific wavelength that you need. For example, if you work with Alexa is a kind of probe that you have at least 3 different wavelengths that you can excite. But if you work with Alexa 488, then you need this wavelength. Then you need the monochromator to select that specific wavelength that you need. This is a monochromator. The monochromator just take the source. The source you have the... it stopped to work. You have the source. In the source, you have the on a full spectrum, you have, or full or one specific interval of wavelengths that we can work. Then you need the monochromator just to select the wavelength that is important for you, and this go to your sample. Then after go to your sample, like I showed to you now, you have the excitation and the emission. But each probe emission in a specific wavelength. Then you need have another monochromator, just select to filter that specific wavelength that you need. For this, that you have one monochromator to excitation, another monochromator to the emission. Then this emitted wavelength, you have a photomultiplicator because you need the signal is very low. Then you use one artifact that multiply that signal that you have, that is the photomultiplier. then you can obtain your signal. Then here you have an example between the excitation and the emission to one aleatory probe. Then the machine basically is this, the setup that you have. Here now I start to discuss about some probes that you can use in the focus of proteins. Then you have the intrinsic fluorescence. That intrinsic is something that is from on the material, that you don't need to add one extra probe. Proteins has the capacity of, if you excitation with some specific wavelength, it can return some energy for you, that you can excitation, emission, light naturally from the proteins. It's like that protein is a kind of probe too. Then you have the 3 specific amino acids that can do this: the tryptophan, tyrosine, phenylalanine. The best is the tryptophan. The best I can say about that, you can see the more intense the effect. The tryptophan, you have the absorbance in 280 is very near to the tyrosine too. Then when you excite you do the excitation to the tryptophan, if you have some tyrosine at the same time excite it too. The phenylalanine is a little bit different, the wavelength. In the emission is different wavelengths. For example, to the tyrosine, the emission in 348, to the tyrosine in 303, to the phenylalanine in 282. Then you have the quant effect that I said. The quant effect is directly to the how intense you can observe that. For this, that the tyrosine is the best because you have a quant effect. For example, is the 50% more than the tyrosine. If you compare with the phenylalanine, is much high the effect. Then for this, that effect is with the intrinsic fluorescence for phenylalanine is very low compared with the tyrosine. Then the fluorescence specific to the proteins, we can correlate it with the fold of the protein. Why with the fold? Yesterday, you learn a lot about the fold of the protein, the organization of the amino acids, but you need to have in mind, if you increase the temperature, you have some limit to maintain that the fold or the structure constant. After some time, the protein starts to denaturation. But if the protein starts to denaturation, these probes, natural probes, the tyrosine or tryptophan, modify the position in the structure. Then this interfere directly in the emission of the light when you do the excitation in the emission of the light. Why? Because if you have some amino acid that is internal of your structure, if you start the denaturation, this amino acid go to the out, this can suffer effect with the solvent. And the solvent is one element that you need to look, because the solvents could deplete your emission. Then what this means? That if you start to look the one specific wavelength, and you change the temperature, you can see how the effect of the temperature to the emission of your fluorescence. Then you can correlate this with the structure of your protein. Let's see this more in details. Then here I just explain I show the properties of each amino acid. That, for example, the tryptophan, like I already said, is the highest absorptive and the high quant effect. Is some descriptions about the tryptophan. I just go. The tyrosine in the phenylalanine is the same. Then it's just the description about the what we have in the table about the characteristics to each amino acid. Then here I think that is the most important for us is about the effect. Then we have here in the top the absorbance, and here is the emission effect of each of these amino acids. In this website, you can look the spectrum to a lot of different probes. Then you can start to think about one experiment. For example, the proteins, you already know what you can expect about the fluorescence. It's about these 3 amino acids. But if you are interested to use another technician, then you can look, for example, in this database, in this library, you look for some specific characteristic of one probe that could help in your experiments. Then here I'm show one example of the use of the intrinsic fluorescence to description one protein. Then here in the top we have the emission. Here you have the intrinsic fluorescence spectrum, the emission that it is a specific protein. Then I evaluate the absorbance between the emission, sorry, between 350, then you can see the emission that I describe here. Then you have the emission of tryptophan is in 348. Okay, then if you look here in the emission, you have here 348 is near the top of the emission. Then when you look for the natural emission of this proton, in the start, in 30 degrees, you have a huge emission, but in the process, with the increase of temperature, you have the decrease of the emission of the fluorescence. Why this is important? This is important because you have some information about how stable is your proton. If you work with Cinetic, you can explore more up each level this protein, this enzyme can works. Then you obtain some experimental evidence about this. Then with the intrinsic fluorescence, you can observe with the increase of the temperature. For example, the fluorescence intensity, you can see that it decreases. You can involve this with the proton structure. You can involve with the fluorescence lifetime of your probe. Then you can explore some information about your targets of study. Then the applications of the fluorescence to the protons permit you explore, for example, if you you can monitor the changes in your proton. You can see some effects of fret in the case, no in this case, but no, in this case too, sorry, because if you look the wavelength of emission to the tryptophan to the others, I will return here, you can see some superposition of emission here, for example, in tyrosine and the filanin. If you look the emission spectrum, you have some superposition, then you can obtain some information about the fret. You can obtain some information about the position of these amino acids in your structure, because you can see if one in the other amino acids can interfere to the other in the distance that you saw this effect. Then this is one of the points that you can explore with the fluorescence to the proteins. But I could say that it's very helpful to see the structure. You can see some amino acids that you can interact to the others in the ligands. Then now I will show another example to use the fluorescence, but when you combine two different probes. No, the intrinsic fluorescence, like in the proteins, but when you use one on probes is some molecules that can absorb emission the light. Then no is natural, but you can add in your system, and specifically use the FACS. What is the FACS? FACS is the fluorescence active cell sorting cells. Okay, but what does it mean? This means that you have one machine that you can, in this setup, you have 4 different lasers. Then you can use 4 probes in the same time. Then you can evaluate dynamic. Why dynamic? Because if I add 2 probes with one proton to the other, I can evaluate about the interaction between one proton to the other. You look for specific probes to see this effect. But this machine, you can explore microscopy studies with fluorescence. Then you can add at least 4 probes. You look at the dynamic in the process. Then you can add 1 probe with a mitochondria, 1 probe with the nucleus, and another in the ribosome. Then you can see the dynamic in the synthesis, the process in the cell. Then you see in real time the process, the organization. Then here, just to you understand how is the setup of this machine, you have one sample. Then here, I will discuss about the sample. Then I just show the system. Then you have a place to inject the sample. This is injected by the machine, obvious. Then in the presence of a laser, this laser can excite this sample in this setup. At least here you have 5 different lasers. But one here is just to the, no, you have lasers to excitation, but you have lasers to just to see the scatter. You have how I say it is, you have a huge organization here, but about the lasers that you can use to excitation, you have 4 lasers, and in this you have another system that you can separate the emission. Then just remember the previous system that I showed you. In the previous, you have a monochromator to select your wavelength, another monochromator to select the emission. Okay? Here is like that you have 4 monochromators to the excitation, and you have 4 monochromators to emission. Then you can select the specific cells that emit that wavelength that you are looking. With the next slide, it will be more clear for you. Then here I just add the channels that we can work. For example, you can excitation using these 4 different wavelengths, but you have filters to select your emission. This is the channel excitation is like the monochromator excitation. Here's the monochromator of the emission. Then look for this example. I am studying, this is a real example that you have 2 proteins. One that is very important for the health, the RAS is a small GTPase that's involved with a lot of cancers. And you look, this is another protein that is a natural effector. Natural effect is a protein that can interact naturally to the another protein. Up this moment, we don't have any probes. I just showed to you the system that I would like to evaluate. I would like to evaluate the interaction between this protein. Okay? Here you can see one other molecule, but this is just a GTP that is the substrate for the RAS. This in the first moment, we don't have any correlation with your interaction that I would like to show. Then, why the RAS is important? The RAS is this protein that we're looking for the interaction with the NOR. Why the motivate to study this? Motivate because the RAS is a protein that is involved with at least 30% of all tumors, cancers that we have in our body. This protein is involved in some degree. For example, if you look for the pancreas, more than 8% of cancers in pancreas is involved in one mutation to this protein. Then it is very important to study. Then if this justifies for you why study this interaction, I go more in the next slide. Then now I just show the RAS. If you look here in my system, we have the NOR and the RAS. Now I describe a little bit about the RAS. The RAS that is important for the health because it's is an GTP. ACE is very important for signal process, to the mitosis, to a lot of process inside the cell. This enzyme is very important, but if you have some mutation in it, it is involved with a lot of cases of cancer in your body. How works the RAS? The RAS is an GTPase enzyme that can bind, is like a switch. What is a switch? Switch is like the light interrupter that you can on and off. When this protein is bind to the GTP is in the on mode, then it is working, but when it's converted to GTP, is in off mode, then it's like that you stop the process. Then if you need emit one message to the cells, then when you start, it binds to GDP. But when the GDP is converted to GDP, this enzyme stops to emit the information. This is natural process of work with an GDP ACE, the RAS, when it works well. But if you have some mutation, in principle involve deposition, the glycosyne 12, 13, or the 61 here, you have one disruption of the function of this enzyme, and you go to the cancer effect. Just to show that this enzyme, when they bind to GDP, it works. When bind to GDP is the natural process, but if you have some mutation, this process could be interfere, and you could just be in the on mode for a lot of time. Then, and just so you know, this RAS enzyme, we have more than 50 years that in the medicine you are trying to study to find some small molecules that could interaction with it to the desacceleration cancer's process, but it's very difficult to find one small molecule. Just 2 years ago, the first molecule was developed by one pharmaceutical company, but just for a specific case of cancer is in the start of studies. Then the studies continue to try to find ways to inhibit this protein. In one of it is use another protein. Then in this case, we have a lot of at least 15 or 20 natural proteins in your body that can bind to the RAS. One of it is the NOR. Then I showed this example. Then here, if you work with the design of protein, you are familiar to identify the area of interaction to one protein. And here, I just select 15 amino acids that is attached very well with this. And my objective in this was to find one modification that I could increase a lot the interaction between these two proteins. Then I select the 15 position. Into this 15 position, I did the 20 most common because I believe that yesterday you saw that we don't have just 20 amino acids, but we have more than 60 in the nature. But the most common in your body is 20. Then I changed each one of these amino acids to the 20 possible amino acids. But I did 2 combinations, 2 or 2 between all these positions. It varied to the 20 possible amino acids. Then I create a library with more than 4,000 mutants of this NOR. For this, is important to use another technician to try to measure this affinity. But it could be impossible to evaluate these 40,000 mutants if you don't have another technician to collaborate to you. Then in case I use the fax, then I just go to the next. But to how I could produce the 40,000 mutants to evaluate with the interaction with RAS. Then you have another way to express proteins. Then you can produce this protein in the membrane of yeast, or you can produce this protein membrane of bacteria you can just produce protein in different ways. One of these ways to produce proteins in the membrane of the yeast, the Saccharomyces, the same that you use to prepare breads, then you can use things in the laboratory to produce proteins. Then you have a specific plasmid that you add inside your yeast cells with the information to the 40,000 mutants. Then I have 40,000 different plasmids. In each one of these plasmids was added inside one cells. We have one process to do this. Then I construct a library with the yeast cell to express the 40,000 different mutants. In each cell, we have a different protein in the membrane. Then is this. Then here I just show. Here you have the yeast cells. The yeast cells express in the membrane the protein of interest. It is here in yellow. Yellow, we have the NOR. By another way, I prepare the RAS. The RAS was prepared in a different technique, just the traditional bacterial expression. Then you express this protein. You have a full protocol to prepare this sample. You add the proteins that you have in the membrane of the yeast, interact with your protein that you express heterologous in bacteria, the RAS. Then you combine these two to interact. Then here was the point. I have 40,000 different NORs to interact with my RAS. And in this experiment, I did the RAS wild type that we don't have any mutation, but I did the experiment with RAS involved with the cancer. For example, the G12 is a mutant. Then in this experiment, I add the NOR to interact with RAS, but the different combinations, just the NOR wild type with the RAS wild type that we don't have any disease. I combine the NOR library, then one pool with the 4,000 mutants with the RAS wild type, but I did the same with RAS mutant version. There's a lot of variables. But why I'm talking about all this? Because we can attach some probes, that's the fluorescence molecule attached to the protein. For example, the CMYC is a specific protein that during the design of this protein, it can bind to a specific area in the NOR. Then I have one protein that I have one fluorescence probe that I attach to my library. Then I can, if I look the signal to the PE, this signal is correlated to the expression of the library, okay? But I attach another probe with the RAS. Here it's like a streptavidin, another protein attached to the Alexa. Alexa is another probe. Then the probe that was attached to the NOR, I could obtain information about the expression to the protein. In another probe that was attached to the RAS, I could obtain information about the interaction. Then in this experiment, I could obtain in the same time information about how stable is my protein, because proteins that are stable mean that in the medium you have a lot of this protein, then it binds a lot with your label. And in the same time, I can obtain information about the expression, the interaction to this protein. Then, now I return to the facts, because now makes sense to use this technique. And then here I just show the emission to each one of these problems, here to the Alexa, here to PE. Then you can see that you have a little superposition between the emission to the Alexa 4080 in the PE, but this isn't enough to interfere in our experiment, because in this experiment the target was to see each probe, to obtain information about the distance. Then this a little bit flat effect here didn't interfere, in the analysis. If you would like to to work to see how the emission to one probe to this different, you can use this website from this company, then you can see how each probe emit. This is very important when you are designing your experiment, you need to look, because when I started this experiment, another probe was much better, and I could use a different setup in the experimental, but the superposition to the probes was very high. Then I couldn't use that probe. Then sometimes you need to change your experiments depending of the technician is direct correlated to the powerful to each probe. And then you need to design the problem depend of this. Then in the case the Alexia, we have an excitation 493, emission in 59, and the PE is the excitation in 5660, the emission in 57.4. Then for this that we have it is little of superposition here. Then here I just show all the steps of this experiment from the design of the gene until here when I do the experiment. Then I do the experiment, I show the emission specters for you, and this information is used to the next steps experiment. Then here I just show again the experimental setup that I used. Then you can see the two labels, and now the result when you use the fax. Then I had my sample now, like I showed you before, then this sample cross the system, then you have the specific probes, each one be excited by a different wavelength, but the emission, then you can filter this. The important in using this machine that I can split the cells that I'm doing the analysis. This is very important because then you can do two things in these setups. You can analyze about the interaction and you can obtain the cells that you did the interaction. Then with this you can, at this moment, I don't know which mutant you have a very good interaction. For this that you can use this setup, then you can split, sort the sample. Then you can obtain your cells that you have a huge interaction. Then you can grow the cells, extract the DNA, send to NGS, and do the identification to each mutant. Because you are working in the order of 4,000. You know, it's just the do an Sanger sequence to obtain. We can do this because you have a huge number of sequences, then you need to do the next generation sequence process. Here, I showed you just the one emission. When you look the Alexia, then you can see the cells. Then if I inject the sample in this machine, you can see in the screen one spectrum like this. You can see the emission to this probe. Then you can see a lot. Each point here is one cell that is emitting light for you in that specific wavelength. Then this is the one experimental control that I did here. And something delayed here. Yeah, then at this moment, I would like that you just see that for each cells, you can see one emission light. When I look for another probe, here I saw the PE that was attached to the, don't do any confusion. Then you have here the PE that is attached to the NOR. When I look the spectrum of PE, I could see this to the Y-type sample. Y-type sample is just the sample without any mutation. Then this was the emission by the Y-type. Then natural, you have this emission. Here, I'm looking just to another probe that is attached to the RAS. Then you can see it here is another, and then each one of these dots here correspond to the protein that is attached to your protein. In this another is the real experiment because you can see the buff labels in the same time. Then here I am showing to you the Y type. What I talk about Y type is the NOR without the library, is the original NOR with the RAS. How these two probes, how is the spectrum of fluorescence to these two probes? Is this? It's like that you have one combination to the two probes. But when you look for the library, you can see a dispersion dots here. What this means? This means that to each one of these is a different mutant. If I correlate the X here, here you have the same affinity to the Y type. In this, if you delimitate some area. Here, you have the same affinity to the Y type, because here's the Y type. But if you compare it with the library, all the cells that are here in P3 has the same affinity to the Y type. This is the point. All the cells here is the original. If you have in your library some cells that have the same interaction in this gate, this means that the cells that are here has the same affinity to the Y type. But if you find some cells in high here, this means that these cells has a high affinity compared to the Y type. But if you see here some dots that are in blue, this dot has the slight low affinity compared to the Y type. And if you look at the P6, the cells has very low compared to the Y type. Then with this, you obtain information about the affinity to the protein in loco. Why this information is important, I just change here to, this is more clear to see. Then what I have here, I have the NOR library, here the NOR Y type. In the NOR library, you have a lot of different cells. Then for this, they have a spread of the interaction. Just to you understand, I didn't explore this, but if you look in the X, you have information about the expression because it's correlated to the PE. If you remember when I showed you the control, I showed here you have information about the expression because expression is correlated to the NOR. Okay? In here, in the, you have information about the RAS in the another axis. Down here, information about the binding, here information about the expression. When you go, we can compare the two with informations from the fluorescence. Then I can select each one of these gates. If I take this position, we have all cells with the same expression, but with the differences in the affinity, because the affinity change to different populations. Then if you sort each of these populations, grow the cells, extract the DNA, and after you sequence this, you can identify which was specific the cells that you have more or less affinity, then you can confirm each mutants are best or no in your study. This is take a lot of work, it is a lot of energy, then you need has a very good target to use this, but when you talk about some target to the health, to the cancer, this is very justified to use this all this setup. Then I just here I'm just illustrate one of the results to the mutations. What information I obtain is when I select the gates. Like I said to you, I have the one gate in the high affinity, one with the Y type, slight low affinity. Then with this, I could identify to each mutation position the amino acids that I found, and the colors mean what? Where you have the green mean that the mutation was very present in that gate. The red is like deplete. Then I could see that we have some positions that you could find best in one group than in the other. And why this was important? Because one another technician that I talk in 5 minutes, I don't know. But in the next slide, I won't continue because I read complete 1 hour. But you could explore another technician to use the fluorescence to calculate the affinity between the two proteins. Then I will conclude my presentation here because I read explores 2 possibilities of use to the fluorescence. If you would like to see about the affinity, the KD, we can discuss this during the day. But I just would like to show 2 different techniques that you can use the fluorescence to explore your targets, to understand about the affinity, the interaction to the proteins, and be free if you'd like to do any questions. Finish here. Yeah. Okay. This is just, yeah. I won't explore this, but you could use the fluorescence to calculate the KD affinity, use the fluorescence too. It's just this. But I'll be here during all the day if you need to do any questions or now. You can do the questions in Portuguese. You don't need to do the questions in English. True? Okay. Let's go to the coffee break.